Thank you, Alex. That was wonderful. Yes, let me introduce Erin Angelini, who is a staff scientist at the Institute and is one of the co-first authors of this work. And I thought it would make a lot of sense for her to come and actually talk about part of the work. So I'm going to do 10 minutes. She's going to do 20 minutes. We're trying to leave 15 minutes for discussion at the end because that's really why we're here. And I'm really excited to start the talk. I wanted to acknowledge all the other people who are part of this work because we do everything in a team way, hence our team talk. But therefore, we also have a lot of people doing things interchangeably as we collect data, do analysis, and we will not show all of the work of all these people, just a subset. So thank you very much, and I'll start. Just wanted to make sure you guys got to meet Erin. Okay, Erin and I are going to talk to you about a dynamical systems framework of how to get at state transitions from morphology-based sort of data. And this is a beautiful echo chamber. It's also a beautiful room. And I actually love giving talks these days because I feel like there's a lot of echoing happening. And so there's a really beautiful arc that goes from Chase to Margaret to me and on for the rest of the day. And hopefully Erin and I will give you guys some food for thought that is different from the food for thought that you've been given by the previous two speakers. So first, just really quickly, there is, of course, momentum to build community around doing cell biology in this big data way and taking what we can get from images. And so there are two meetings I wanted to put up there if any of you are interested. And I'm just going to quickly zoom in on this one because it was too small to read. Depending on where you land, I think either or both of these might be exciting to you. One is being organized in part by the Allen Institute, and one is being organized in part by me. So I thought I would be shamelessly put a plug to continue this community building in this space. So obviously, we are in an era of large-scale cell image data sets that are popping up everywhere, and thinking about how to use them is a question. We want to make sense of them through quantitative image data science methods. But if we think about what these data are, we also need to ask, what is it that we need to know a cell? And so we're going at this by saying, well, what we need to know is what state a cell is in. And if we do this in a data-driven way, what we mean is what are the range of values for each observable that we have that describes a cell in some relatively stable state, where we define cell observable as anything we can observe or measure or see about the cell. And then we'd like to understand how and why a cell changes state, which is the dynamics of how these observables change as cell leave one state and settle into a new state. And the theme of feedbacks, of course, is really key in all of that. So Julie and I wrote a perspective last year. It's 17 pages, way more than you might want on some of these topics. But in it, we sum it up in like 2 minutes. We really believe that, of course, now is the time to update the concept of cell state to be a holistic cell state, where we bring quantitative spatiotemporal observables together with molecular observables. In the biology community that is beyond these walls and this set of people, a lot of what is being done in biology is believed to be completely encompassed by doing a lot of molecular representations and thinking about molecular cell state. But cell state, of course, traditionally came from what cells look like, where they're located, what they do, and then what molecules are in them. And so we want to define holistic cell state as a combination of aspects of each of these. So what we then therefore believe is that what we need to understand or to know a cell is to understand the bidirectional interplay between these categories of observables in time. So Margaret's words of the feedback. Of course, this is not just for observables, and it's not just for categories. It's a continuum, and it could be a vector of all observables that you can imagine, and then they all can have their feedback. And this sort of diagram is one that you see a lot in systems biology. You see it in neural dynamics, and really the only and main point we're making, which is a point that is somewhat moot to this room, but I'm still going to make it, is let's do that, thinking about all of the observables coming together, and let's think about integrating. It's this sort of feedback and these sorts of diagrams that get us towards thinking in a dynamical systems framework way about holistic cell state. And it's, of course, also the reason that we need quantitative observables and need all of the experimentalists and the descriptive modelers to then, actually figure out what should be pulled out. And then just an important point, we also are going to wrestle with the fact that we're never going to have a complete set of observables, and we probably don't need one. So we need to think about how to approach this with sparse data and which sparse data we want. So if we want to get at mechanisms underlying normal cell states and disease and anything you can imagine, we have to be able to integrate across categories of observables. And we have to do that via quantitative frameworks, some of which you've already heard about this morning. We're very, very good now in the biology space at this moment in time at molecular representations via bulk or single-cell genomics. We are less good at organizational representations, such as if you think about the progression of prognosis of cancer and the pathologist and the fact that they just look at sort of what the nuclei do. That is not actually harnessed in an interpretable way in such a clear way yet. And that's a really simple one. So really, how can we move towards holistic cell representations? And we can have a debate about whether those do or don't or should or shouldn't include voxel-based versus non-voxel-based approaches. And then again, just to say holistic does not mean complete. And I think you'll hear about that. So since we're at a meeting, we're also talking about how we take advantage of this moment in time. How does AI fit in? Well, if we want to think about this very long vector of all sorts of observables and how they interconnect with each other, of course, we're going to get beyond what our feeble human brain can sort of track. And we want to have pattern detectors that help us identify all of that. And we also want to do that because we want to do, if we can, wise and smart dimensional reduction in order to get these holistic cell representations. We focus in a lot on generative AI. I think you've been hearing this as well. We really like to decode our latent space. We like to understand what it is that is the pattern that is being seen. A, in order to get rid of noise or microscope stuff or who knows what, and B, because it actually teaches us something. And it is really that beautiful creativity that we can all play with that particular sort of interface of pattern seeing, deciding whether we've seen the pattern we want, and then going further. That is, I think, a really exciting moment for us. Our goal is predictive understanding, not black box prediction alone. I don't have to say that in this room, but I do have to say that in many rooms that I speak in. So. At the Allen Institute, the Allen Institute for Cell Science just launched its second 10-year initiative, which we call Cellscapes. And it aims to answer the question, along with the community, about how do cells organize themselves across scales to form complex cell communities and tissues. So in the first 10 years at the Allen Institute, we studied human-induced pluripotent stem cells as our model system, grown in these colonies that are basically sort of a 2D sheet, but of course with 3D cells. And so you can see here nuclei of these cells growing and dividing. And as we move into cellscapes, we're really moving more into 3D so that we can ask questions of morphogenesis. So these same cells, if we add just a little bit of methylgel to the matrix or do other things that Margaret can tell you at length about, form these beautiful luminoids, which are a single cell layer, 3D structure. And so we can ask questions about how they form, what they do, how they work, how they are maintained. This picture, remember, they're just a single layer. So there's no cells on the inside, there's lumen. So our goal is to achieve predictive understanding of cell state transitions during multicellular morphogenesis. Our Cellscapes initiative, so many people are doing this, and we all should be. We have a very particular targeted set of data, model systems, and approaches that we're going to use so that we can try to create instances, along with others in this room, of how the larger field, can go at doing this. So we have 4 questions that we focus on. One is, how do cells respond to environmental changes? You'll hear a lot more about that. Another is, how do cells form complex 3D structures, where we're focusing in on the 2D colonies to 3D luminoids, where we don't have a significant gene expression state change. It's all morphogenesis. Then, how do cells differentiate and move, which of course would have laid upon it all of the developmental programs that underlie something like these cells then undergoing an EMT. And then we have our synthoids, where we can ask whether we can program cell behavior. Our goal is predictive understanding of multiscale multicellular morphogenesis. And so, of course, we need to really think about how we bring in explanatory and mechanistic models and theory. And Issa Nazark just joined us 41 days ago as our new director of explanatory modeling. And together with Mateus Viana, who is our director of descriptive modeling, they're really going to work on bringing together top-down and bottom-up approaches to try to iterate with our experiments. We also really aim to understand, and what we want to be able to predict, we also want to be able to test experimentally. And so we have a project, and that's really the project that we're heading toward in our 10-year timeframe, where we'd be able to build to understand. We take these luminoids, we start to create cell states in them, start to create other activities in them, and can we predict what they would do if we've understood the system well? At the core of all of this, the thing that ties together our data and our modeling is, of course, this question of appropriate cell representations depending on the question. So today, we're going to focus on endothelial cells under flow. This is a 2D proof of concept model system. It is not our luminoid system. And that's because what we wanted to try to do is figure out how do we go from dynamic images of what our cells do as they do a state transition to landscapes of these states in a quantitative way, perhaps not in a segmentation-based manner, as you will hear. And doing that in our 3D crazy system that does all of this other stuff, that was probably not this place to start. So we started with our endothelial cells. So our human-induced pluripotent stem cells, they're cells that, are scales that you get from the skin that get retransformed to be pluripotent. And they can then go forward and differentiate into many different cell types, one of which we did is the endothelial cell. So we can do that. Here's a bright field image of what these cells, once they're endothelial cells, look like. And here's also magged up a little bit more. The cell line that we're going to focus in on a lot is expressing VE-Cadherin, which marks the cell-cell contacts between these cells and is also a really good marker for seeing the cells. So endothelial cells, of course, are the cells that you have in your blood vessels. And so they really like to respond and behave normally under flow. So we grow these cells in these EBIDI flow chambers, and we have flow that's going over the cells. And under no shear stress, they have a mixed alignment. And then when you put them under low shear stress, they have a parallel alignment. And in a very interesting way, when they're under high shear stress, they have a perpendicular alignment against the flow, which you see at the bottom. So we can image these cells in bright field, and we can have our VE-Cadherin cell line. Oops. channel. And so here's what this looks like if we image these cells for multiple days. And you can see that initially the cells are starting to align because they started in this mixed orientation. I'm going to tell you a lot more about it. We image these in these very large sort of 6-stitched fields of view so that we really have the continuity data of also what the environment is doing. So we have our byte field, and we take everything as z-stack. So at the bottom, you can see what a z-stack of VE-Cadherin looks like. These cells are 3D, of course, and the third dimension does matter. Erin will tell you when. But primarily, we can sort of focus in on them being 2D. And the way that we do that is, in the case of the byte field, we actually can hardly see the cells, which makes sense. But if we do a standard deviation projection, we get all sorts of rich features out. And so we're really focusing on that. And that's used for segmentation-free analysis and integration. And then, of course, for the VE-Cadherin, we can use that if we want to do more standard things like segmenting the cells, tracking them, getting their shape features, and so on, which we have also done. So these cells start this way. And the early response, the first 18 hours, they align. Oops, once they have aligned, I went too fast, but that's okay. They start moving upstream, and they are aligned this way. And they also have this puncta at the back end that is related to the sensing and responding to the flow. And then at some point, they get overgrown and overgrown some more. And so what we're going to do today is really focus in on that steady state period where they're not overgrown. I already showed you what these cells look like under low shear stress. And so here's also a zoom in of the puncta. But under high shear stress, it's actually really quite beautiful. These cells don't move quite as much. They are still very aligned in a perpendicular fashion, and they do have movement. So their morphology is elongated and perpendicular. The migration is many directions and often perpendicular to the flow. And they no longer have these puncta. So with this type of data at these steady states, we want to ask or talk about today, how do we use this microscopy data to quantitatively define cell states and to generate a dynamic landscape of cell states? Erin?
All right, thank you, Susan, for that setup. So where do we start when we want to think about how do we quantitatively define cell states? Where we start is feature extraction. What are the quantitative observables that we can use to define our system? We could proceed with the classical image analysis pipeline, where we would segment our images and track single cells. And then we would get a sort of hand-selected set of observations, or I guess measurements from our segmentation, such as orientation, shape, angle of migration. The fun thing is that I'm in an audience or speaking to an audience of people that understand that there are a lot of challenges with this approach, and it is not as always straightforward a pipeline as we'd like it to be. The other thing I do want to highlight as a challenge for this approach is that there's often a bias in our human identified choice of observables. So we like to look at our images and say, what are our identifiable axes of variation that we think are important to the biological system? However, this gets at a much more kind of broader conceptual question, is what are the necessary and sufficient observables for a complete characterization of our system? And what do we mean by completeness? So given all these sort of challenges and, I don't know, philosophical ambiguities, we pushed ourselves to ask, can we instead learn the features in an unsupervised manner that are the important features for our biological system at hand? By learning, I mean implementing a machine or deep learning, aka AI, approach for unsupervised feature extraction. What would we want from such a framework? Well, we are looking for cell representations, so these feature-based representations that are complete in some ambiguous sense that I will kind of discuss more in a bit. They're compact in that we want to do some sort of dimensional reduction, and they're interpretable in that we don't want to just get a vector of numbers and we don't really care what they mean. We care very much what each of those numbers represent in terms of biological understanding. One thing we also want to think about is how we can choose quantitative anchors by which we can integrate data across multiple categories of observables. For example, we want to be able to compare morphological observables with molecular observables, such as transcription factor expression. One thing I do want to emphasize and kind of plant the seed for future thought is when we say unsupervised, we do not mean taking a naive or uninformed approach to our selection of model architecture and how we train it. And I think this is something that Margaret's talk has set us up nicely to think about and really have this discussion with the people that are here in this workshop. So what do we do for our data to obtain biologically interpretable features from our data, we want to be able to extract information from these images on a similar spatial scale to that of a single cell, but again, in a segmentation and tracking-free manner, instead using deep learning in place of that manual selection of observables. Regarding the question of the spatial scale, we may take many small crops of our 2D images about the average length of our endothelial cells, and then we crop the bright field images to those small crops at the same location that we crop the VE-Cadherin GFP. Regarding the deep learning model framework, we settled on a class of diffusion models called diffusion autoencoders, or DIFF-AEs for short. Some of you might be familiar with text-to-image diffusion models, in which a vector embedding of a text prompt that you give guides the diffusion model to generate a specific picture depicting your text input. We're using a similar architecture, except that the embedded text prompts are replaced with embeddings of our bright field crops. And the range of images that the diffusion model is trained to reconstruct are the VE-Cadherin crops. The idea is that when these two networks, the semantic encoder and the diffusion model, are trained concurrently, the semantic encoder should learn the high-level features of the bright field images that best inform the reconstruction of VE-Cadherin. This is how we are using deep learning to get us toward compact and interpretable cell observables. I want to highlight the fact that once we have a trained model of this form, we only need bright fields to obtain these semantic features. So what we can then do to get an interpretability is we can condition the diffusion model's inference process, in which we repeatedly denoise a starting quote-unquote image of pure Gaussian noise to generate a synthetic VE-Cadherin image representing those semantic features learned from the bright field. Before we go into the next section of the talk, I want to give a very honest and transparent disclaimer. That is that the results that we show on the following slides are not quite as ready as we had hoped, because science happens. Particularly in preparation for turning this project into a manuscript, we did our due diligence and did the proper quantitative controls with our model training, and realized we need to adjust some key aspects of how we were setting up our model training. What I do want to emphasize for this talk and for this audience is that our conceptual approach to how we analyze those features that we get from the model is sound and is what we wanted to highlight for this audience. Specifically, we don't expect a significant change in our qualitative interpretation of the results, just in the actual quantitative nuances of the features that we get. With that in mind, we've established this feature extraction pipeline by which we get a collection of vector embeddings from these crops of our bright field images. And we're going to be focusing on our 3 baseline data sets that Susan showed in her introduction, cells under low and high shear stress, and a no shear stress control data set. To better understand what these latent features represent and to perform further dimension reduction, we can perform PCA on this collection of vectors. Doing so, we find that the top 3 principal components explain over 98% of the variance within these 3 data sets, and that our 3 different data sets are well separated in our principal component space. Focusing on the first dimension, what does it represent? We need to get back to the interpretability part. And this is where the generative aspect of the diffusion autoencoder comes into play. Recall that we can condition that image generation process on a given latent vector. Doing this, we can perform what we call a latent walk by generating a set of synthetic images corresponding to the coordinates that we get by walking along each of our principal component axes. As we do this and walk along the axis, we see that an increasing value of principal component 1 captures the reorientation of cells going from perpendicular to parallel to the direction of flow that we see going from our high shear stress condition to our low shear stress condition. As for PC2 and PC3, we have preliminary results that indicate that they're density, but this is part of the nuances where we went back and said, I think we need to retrain and rethink how we're training our model. So TBD before we can finalize that statement. But returning to our motivating questions, once we have done feature extraction, how do we do dynamics? In particular, we are thinking about how to get to a dynamical systems representation for our cell state observables, because it gives us a conceptual link to establish theories for non-equilibrium steady states and generalized landscapes for biological systems that have been more established in the biochemical signaling and network world. Oh, and then I wanted to point out that we got a nice flavor of that yesterday afternoon with one of our talks. So how do we get there? Consider a single crop for a bright field projected at a single point in time At this point, we've established our feature extraction process for these crops. If we advance the frame to the next time point, take a crop of that in stable location and map it into our feature space, we now have a displacement vector for a single crop location over time. Repeating this process for many crop locations and all time points in a given data set, we get a collection of these displacement vectors. We can then take these samples and perform some sort of regression. We settled on kernel regression to estimate the function f that defines the underlying dynamical system. In other words, we are tackling an inverse problem, not unsimilar to the one that Chase set up this morning in his talk. In particular, I'm an applied mathematician, so I like to be clear about my assumptions. We formulate this inverse problem under the assumption that we have a time homogeneous system that is parameterized by shear stress as a variable. Pictured here are the vector fields over our principal component space that we get from performing this inverse problem estimation on our data at low and high shear stress specifically. So some of you might be familiar with the Waddington landscape metaphor, in which we have stable fate trajectories being the values of the system. In the language of dynamical systems, these values as stable trajectories correspond to stable cell states of our dynamical system. So this is where we can think about stable fixed points as stable cell states. And this is what this looks like for our low and high shear stress data sets. And recall that we can take a point in our latent space and use it to guide the image generation process of our diffusion model to reconstruct the VE-Cadherin signal that the model has encoded as representative of that point in the latent space. As such, we can get several VE-Cadherin reconstructions for these stable fixed points that we see at low and high shear stress. And we find that the generated images for each of these stable states are exactly what we'd expect to see in terms of morphology, orientation, elongation, etc. So far, we've analyzed the dynamics separately for the data at low shear stress and high shear stress to identify our stable cell states in our diffusion autoencoder cell representation space. However, to get at the question of cell state transitions, we need to ask, or part of the question we want to ask, is how the dynamics at low shear stress become the dynamics at high shear stress and vice versa. That is to say, what do the dynamics, what is the underlying structure of our system, what does it look like at shear stress levels intermediate to low and high shear stress? To get a better visual picture of what this means, let us think about our dynamics as encoding some sort of landscape over our state space, where the stable fixed points, again, the heuristic is that these are basins or valleys of our landscape. And so pictured here is what it might look like along the first principal component axis. So how do we get there? We look at the data. When we look at the example of our movies at 9 dynes per centimeter squared, we see a resemblance to our low shear stress steady state. And on the other end, when we look at our cells under 15 dynes centimeters squared, we show resemblance to the high shear stress steady state. Where we see something really interesting, I think, is at the intermediate value of 12 dynes, where the morphology, alignment, migration, and signaling behavior in our cells is all over the place compared to what we see kind of uniformly at low and high shear stress. Conceptually, we're thinking about 2 possible scenarios in terms of our dynamics and feature space. One is where we have a continuously moving basin, aka stable state, going from the one that we see at low shear stress to what we see at high shear stress. So our intermediate phase is monostable. Another scenario is where we have the appearance and disappearance of our 2 basins as we go from low to high and vice versa. In this case, our intermediate phase is multistable. This is what mathematicians would call a saddle node bifurcation, and what physicists might term a first order phase transition. What we find when we actually quantify the feature dynamics at 12 dynes per centimeter squared, we find dynamics, so underlying flow field and stable fixed points, and as well as a distribution of data that suggests being near that critical transition of a saddle node bifurcation, where we have multi-stability, but we have one stable fixed point that is much more stable proportionally to the other. Returning to the disclaimer that I gave at the beginning of my section, this is somewhere where we expect the core qualitative result of having this critical transition will not change even as we update the particulars of our feature. And that this is something we believe we see in the actual data of the cells we see. So what we've done so far is map out the underlying structure of our system along the axis of shear stress, building sort of bifurcation diagram of the process. Going continuously, however, from one dynamic steady state to another is not something we can do experimentally. We can't just have the cells go to one steady state and then so finely tune our shear stress to keep it in that basin. Instead, we can do something where we change the level of shear stress mid-experiment and see how our cells respond. When we go from low shear stress to high shear stress, what we see, as in this movie, is that the cells stop migrating and they very quickly reorient their angle of alignment and migration, settling into that high shear stress steady state that we saw before. Similarly, when we go from high shear stress and then switch the cells to being under low shear stress, we again see a quick readjustment of their morphology and their migration and signaling behaviors according to that shear stress level. So what we're trying to think about is how this fits quantitatively into our picture of looking at just the long-term dynamics instead of the transient dynamics. Another way we can ask this is how do we quantitatively define this sort of state transition that the cells actually undergo as our shear stress has changed? How does this fit into our conceptual picture? We're asking the question and thinking about, can these dynamics be simply described as a sort of jump relaxation process, where we have the cells kind of hopping between the landscapes that we've quantified from our single shear stress data sets at steady state? Do we see hysteresis in our system? Do we see any memory of previous flow conditions? If so, is it a sort of hysteresis that can be sufficiently explained by the hypothesized cell node bifurcation, or are there additional memory effects coming from possible unobserved state variables? So this is getting back to that question of what does it mean when we say a complete representation? One version that we can think of as completeness is completeness as measured by memorylessness, or having a truly Markovian system, that when we have all of our features at one time point, we can predict the probability of transition at the next time point. If so, what might these biological observables be? We're thinking about if we do see unobserved memory effects, or we observe memory effects from unobserved state variables, we're thinking about this be related to the VE-Cadherin puncta and cell signaling. You might say, why are you already thinking about that? Why are we thinking about the puncta? Well, it was a conscious choice to exclude that aspect of variation from our model, in that we did not expect our puncta VE-Cadherin to be detectable in the bright field. So we knew that we might need additional measurements for those. I think this, again, ties into really nicely what Margaret was saying in terms of what images can predict other structures and where do we have breakdown in that predictability. So why did we make this conscious decision to exclude something that we thought might be very important to our system? This goes back to the question of how to anchor integration across observables. So far, we have only shown you data from experiments where we collect VE-Cadherin and bright field. However, in order to collect observables from other aspects of our biological system, we will necessarily need to target things other than VE-Cadherin. The advantage is that what we have is that for all of our experiments, we always collect bright field. How do we do this? How do we do this integration without the need to create endless pairs of dual or triple cell lines? So still thinking about small image crops, we can obtain whatever quantitative observables from our images from our new experiments that we want. For example, this immunofluorescence experiment looking at a SNAP1. Well, how do we get morphological observables that we could relate to, that we could have obtained from VE-Cadherin? We can take our corresponding bright field crop that we always collect. We can pass it through our semantic encoder and get a common reference space of our features, our cell morphological observables. And then we can use this diffusion autoencoder to get a reconstruction of what the VE-Cadherin might look like for that example bright field. And so this is an example of how we're thinking about doing integration of morphological and molecular observables. At least that was the intention for why we chose bright field as our anchor for featurization. This gets us back to the point, though, that we really want to drive home, which is that using AI for feature extraction in this manner is not something that we want to do naively. There are conscious choices that we need to make and have made to set up our deep learning framework for success, so to speak. By success, I mean obtaining a latent feature space that does not just give us an accurate representation of our images, but also a biologically informed one. Again, this relates back to the question of completeness of observables in terms of what is both necessary and sufficient to describe the range of biology that we see by eye. One decision that we need to make is what data we use to train our model. So for example, do we want to include the cell overgrowth phase that we see in our data in our training set, or do we want to control for this aspect of image variation by excluding it from training? We also need to think carefully about our pre-processing steps. For example, when we think about cell height, which does change as cells get denser over time, we need to think about what range of Z-slices are appropriate to capture that range of cell height. We also want to think about how we normalize our images. There are standards for normalization used in the AI analysis community, but do these transfer over to our brachial images, to our live cell images? Is this something that we just want to naively continue using? We also want to think about what spatial scale of the image crop is too small or too large. So how much of a single cell versus its environment do we need to include in our crop to get the right sort of featurization? More broadly, we need to just rethink the standard practices of the general AI community, be careful about which practices are sufficient for our scientific needs, and readjust accordingly. In sum, this is an emerging field in which we have not established a collective understanding of our ground truths. We cannot cut corners in terms of performing controls and understanding what our model is actually doing until we have that solid understanding of what our controls should be and how these models behave. If our goal is to have interpretability, we don't want to just keep on building on something that was trained naively. So this is something where I wanted to bring this back to having the opportunity to give this talk at NITMB gives us a really unique opportunity to present this side of the work. So instead of showing you the tidy final product, we get to tell the full story, because the people in attendance at this workshop are also the people who are thinking deeply about these questions. So if this is a conversation you want to have more in depth, ask those questions now, and I will also be giving a presentation at the poster session this afternoon. Finally, I'd like to thank our entire cell science team, as well as two of our close partners on this given project, Julie Theriault and Jin Wong, who all contributed to parts of this talk. So with that, I'll be done the thank you slide.
Questions? Yeah, really nice talk. I was wondering, based on the models that you have, do we know the biophysics of why cells align differently? And can we somehow use the physics as a way to add that additional constraint on the models to make them?
I'll let you.
Okay.
Take first.
So endothelial cells and their behavior is well established, and there are cell lines where people look at this, and in those cell lines, people have observed all sorts of really interesting, even sort of this reorientation to flow. But the most common cell line, the HUVECs, they actually don't move as much. So there's a wonderful paper out there. I'm about to show you one piece of data I asked Aaron to put in here as like the one thing that I want to show you guys, because there's in that cell line, you can go look at the paper. They were really looking at and starting to think about the differences in internal polarity of the cells, which of course would feed back into the biophysics. And of course, in their situation and with their data, and I couldn't quite tell fully from their data, but there's really not that movement. So we have the system, our endothelial cells, they're baby endothelial cells, like they're differentiated from the stem cells. So when we talk to people like Martin Schwartz, who's like a big endothelial expert, he's like, oh yeah, they're their own special thing. That is perfectly fine for us. It's a model system, but in the context of how that then fits into the biophysics of what is established, it's not the same. So we have been playing around with some of that. It's not going to be in our first paper. And actually, before, I'm going to show you the cool little bit of data in a second. But I was going to say, one of the challenges of the Allen Institute is that, when I came from Irvine, I actually, left my whole, research program to switch over. Isan is going through that right now. But we all come together and, take this big cruise ship on this ride. Along the way, we decide which islands we stop at and which ones we don't. And endothelial cells is our proof of concept system. And we're going to end it at the end of this paper. Sorry, Margaret. But there's so much that we want to do. So we're actually going to seed what we're calling the endo party, and try to get others who are interested in following up, maybe with our system or our approaches, even in these cells, to do a lot of the things that we've already started doing and tools that we have built that we won't get the pleasure of continuing with. That said, I will give you one example. That is not the example. Okay. This is our tubulin cell line. for our cells now under low shear stress. And you can see beautifully the polarization of where the MTOC is, and it sits, upstream of the nucleus as the cells move, and the bundles of microtubules. And there's all sorts of wonderful modeling you could do with this sort of data and with the knowledge of the measurements that would come from this. Now, before I show you what happens at high shear stress, make a mental model of what you think should happen when the cells align perpendicular. Where would you expect the microtubule organizing center to be? Where would you expect, if the cells are moving upward, things to be oriented? Everyone have a mental model? Everyone ready? So my mental model was wrong. Hopefully some of your mental models are wiser than mine. So you can see this especially with this. Let's see if I can now do this. With this guy, yeah, he's gonna, when he loops, there's this guy that moves up and down, and you can definitely see that the movement is perpendicular. The MTOC stays right where it was, next to the nucleus. The location of the MTOC is sensed by the flow. The motility is going against that in a perpendicular fashion. And the thing that is polarized is actually the thick bundle of the microtubules, not the location of the MTOC. So for people like me who are classic cell biologists, there's just so much in here that I want to get at. And so just to say that, no, the answer to that was my very long answer, no, but this is why we save 10 minutes for conversation.
It's a really excellent question. I think something I've been trying to grapple with internally throughout this project is, right, I think we can talk about doing AI for feature extraction biology. We don't want to do this naively. We want to do this in a biologically and possibly physically informed way. You can say the same thing, as Chase so well laid out, about system identification. We want to use physical constraints and physical properties, or even just qualitative biological properties that we know about the system, to constrain what we expect our dynamical system to be, and what we might expect the features, what features are important, and how we can constrain that model representation. And so I think in terms of this talk and this project, we focused on the feature extraction part of it for internal reasons. But I think this is something that's I've been mulling on a lot. So.
And now we promised we won't answer every question for 10-minute diatribe.
So I know this is sort of a test problem, but I'm kind of curious about something about how you think about this. Given that the main thing that changes with the flow is the alignment direction of the cells, it makes sense that the major, feature that you would find is the alignment of the cells. But given that you kind of knew that going in for this particular problem, couldn't you argue that just hand selecting the alignment of the cells worked and would have saved you like a lot of computing time?
And so this is exactly where some of our computational controls with the model and trying to disentangle PCs2 and PC3 led us to go back and say that exact question and to say, are there ways that we are still going to, is this an aspect of just this is the main aspect of variation? Yeah. And there aren't things that we're going to pick up from the bright field in this manner? Or is this something that is this something about how we are training this model and what we are including or not including in our processing or exclusion in our data sets that is biasing our model?
The way we thought about it is ideally you would do both. So, but we didn't actually have cell segmentation and tracking until recently. We now have access to those features, which we didn't. So actually, when we set up to do the model, one of the decisions we were going to make is just use VE-Cadherin, and we would get the puncta out. We would set the model up this way. Along the way, we discovered that with this particular architecture, we could do this cool thing of really trying to do integration and go broader. And so if this was a bigger paper that we were going to take another year to do, we could bring all of this together. We're choosing, because of our priorities, to cut the paper basically here, for a little bit. And so that question may not get fully explored, but that is absolutely valid. It's also our saving grace if the model ends up not working. We're going to go back and do that to still have the proof of concept of the paper, which is really how do we get the landscapes from this sort of dynamic data. The model was sort of this bonus that allows us to explore how to try to do this in a segmentation-free manner, in an integratable manner.
I just want one naive question, maybe I missed it. Is that we talk about this feature, what are these features in a biological sense? They're just alignment or?
You mean the features of the model sees or the features we're talking about?
Feature from the model, from the diffusion model. What are those features?
Anything that is visible in the bright field that the model chooses to think matters in order to create the pattern that is the VE-Cadherin.
What is biological meaning of those features?
Yeah. So when we do you want to take that?
I think
When we do the PCA, what we're saying is what the model is seeing is the features that represent in the bright field the alignment of the cells as well as the density of the cells and potentially more. When our model is fully tuned, we will know how far it goes. We could pause and say, hey, those are the only two we need, or three, do we need more? But we would pull more features out than just alignment, maybe angle of alignment, movement, direction, all of these aspects. What the model sees versus what we say we see may not be one-to-one, and that's what we want to explore. We can't tell you the answer of that right now. But this is a simple enough system that we would hope that we can capture all of it. That's sort of the point. I mean, that's sort of like Margaret, you know, when you and Vincenzo were going back and forth, it's like something that is very established enough that you can actually ask these questions about whether you're using the approach to do something new. And then you're going to go into the brave new world of the unknown. So.
Hey, nice talk. I was curious, can you speak to how you think about like variability and fluctuations in this framework? Because you have latent space and you have these clouds, but it's not cells jiggling around, it's your movies are jiggling
Yes.
around latent
Yeah.
space.
So this is something, again, was not in the scope of this talk, but is in the scope of this project. And I unfortunately didn't include these supplementary slides, so I'll chat later. But basically, we're also thinking about, so originally we didn't have segmentation and tracking, and then my wonderful coworker Serge figured out how to get great segmentations out of these noisy images and track the cells. And so now we have those, but we can ask the question of, instead of using this fixed frame, displace the image, we can also ask if we center, say, our crops around the nucleus, and we track the nucleus and crop it, and we look at from time point 1 to time point 2, the crops that are the nucleus, how does that change? So like sort of a cell-centric crop, are those different? You know, in the 5-minute time interval in between frames, is the displacement of a cell so large that the surrounding image is going to be like noticeably different than if we just fixed the location and looked at how the image changed? So excellent question, and something we are thinking about and incorporating this project.
And it looked fine.
Oh yeah, and the preliminary again.
It was fine. It was Julie was obsessed with this. She's like, you guys cannot make statements about cells if you're looking at crops. So we're good.
Yeah.
So I have a question about the saddle node bifurcation.
Mhm.
So you did this dynamical systems analysis. You see this particular type of bifurcation. If I remember correctly, it happened somewhere near the medium stress condition, but not right at. So maybe two questions about like, what would your prediction be if you would tune the stress condition right near the bifurcation? And maybe zooming out a little bit more, could there have been another kind of structure that could interpolate between the low and high stress? And is there, you know, does this particular bifurcation tell you something about how cells are really organizing this way?
Yeah. So I think these are all fantastic questions. Let me think about how to answer them one at a time. So in terms of like, yes, you were hitting on the fact that when we look at this intermediate shear stress level, we're getting at least what we think we see. And then also just to clarify, this second fixed point, it looks like there's no data there because of the opacity. We confirmed that this is not just like a hallucination of the kernel regression. This is actually something where we see a small amount of cells cluster. And so this is why we hypothesize that it's at this very close to critical transition where there's still some remaining stability. In terms of what would happen, what we might think happen as we tune the shear stress, we've gone back, you've hit on a big discussion point internally, especially with our collaborator, Jin Wang, is, right, we can always go back and try and ask the experimentalists, like, can you just get like something in between 12 and 15? And like, could we like map out so continuously this bifurcation diagram? And the tricky thing experimentally here is that the set point of the avidity for actually like how we want to set the shear stress level is not as precise as we'd want it to be to get that sort of fine-grained observables. So when we have this low shear stress range going from 5 to 7 dynes, that's because we set the shear stress at 6 dynes. And the actual measured shear stress on the output is usually, it has quite a bigger range. So in terms of getting super fine grain, we could set it at, say, 10.5, but would that actually just bring us back down to 9 and things like that? And so this was kind of our compromise, is to not just look at low, this 12 dynes per centimeter squared intermediate and high, but try and map this out for 9 and 15 as well, and then maybe see if we can apply some sort of optimal control framework to see if we can interpolate what it might be in between. That was your first question. Could you repeat your second question?
Well, maybe my final question, could there have been another way in which this bifurcation diagram could have looked? Is there something really significant about this particular result?
Yeah, I think an excellent question. And I think, is there something else? Right. Okay. So I think the two scenarios we're thinking about is that we have this continuous transition, like no bifurcation. We just have intermediate, whatever in our feature space, whatever that kind of state is just some sort of intermediate along PC1. Or do we see this multi-stability where we see cells transitioning between these basins? And I think looking at the movies, right, there could be a world in which this is not simply like a mix between high shear stress and low shear stress. And I think this is something where thinking about this feature extraction and where we're going to need to bring in some cell-centric stuff, I think it's going to be interesting because, yeah, I don't think looking at these movies, I think there is something going on.
I think
let's talk about this later. You've touched on like a really critical point.
The hidden variables, the ones that are currently not in our complete set, are going to matter. And we are, if you know, this particular state is right there. But I'm going to tell you that, you know, the way that this looked is Jin says, please, more shear stresses. And Julie says, you have too much faith in the experimentalists. Jin says, no, I mean, please, more now. And Julie says, no, not if we're going to get this. This is literally the. But if you look at that movie, there's a lot of interesting stuff going on that relates to also the cells around each other and what they're doing, and we have not dealt with that properly yet.
Yeah.
So